Hello everyone. In this session, I shall be discussing some revision of enzymes and nitrogen metabolism related aspects that would really help you for preparation of your upcoming exams. Let us first see the classification and nomenclature. Our first session we have detailed description about this particular aspect. See, we need to remember we also discussed there about the mnemonic, how exactly oath lil t or OTH lilt can be the easy mnemonic to remember this classification, which consists of oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases and translocases. Let us see quickly what are the different types. Oxidoreductase. Oxidoreductase means at the same time oxidation of one substrate and reduction of the another substrate takes place. That is called oxidoreductases. That is EC1 enzymes. As we all know, the naming system or the nomenclature of the enzymes has now been following. We are following the IUPMB system of classification. Earlier classifications were adding suffix ASE to the substrate name, and also based on the reaction name and also the reaction and substrate name, like succinic dehydrogenase, like that. But nowadays we are following a very systematic detailed description about the catalytic action of the enzymes based on the IUPMB system of classification. This particular oxidoreductases in IUPMB, it is called enzyme commission number 1. That one refers to the major class oxidoreductases. And other 3 digits also add to this one EC1, that is the specific code according to IUPMB consists of 4 digits. First digit tells us major class, second digit tells us the subclass, and then subclass, and finally the specific systematic name amongst the similar type of enzymes in overall enzyme classification. And the second, coming to the second one, that is transferases. As the name itself is suggesting, transfer of a particular functional group from one substrate to another substrate, converting them into product. For example, methyl group, ethyl group, amino group, and also hydroxyl group, and very importantly the phosphate group, exokinases, phosphates, the transfer of the energy rich and ordinary phosphate groups. That is the main motto of this transferases. You can see here schematic representation. AB are the substrates plus C is the one functional group containing reactant, so that from AB, B is detaching and it is going to take up the side chain or the functional group. So A plus BC, that is the addition of a transfer of the functional group from one substrate to another substrate in giving out the products, that is transferases. Hydrolases, addition of water to the substrates, making them the products. All the digestive enzymes are hydrolases because since the pepsin, trypsin, chymotrypsin, all those enzymes which are framed by the dehydration reaction, so they need hydration to give the products, to dissociate and give the products. And lyases means cleaving, lysing. Okay, here we can see one of the group substrate AB with X and Y they are going to break this bond with X and Y, and they will frame the double bond in the product. That is the meaning of the liases. And isomerases, they are the molecular species of the same enzymes. For example, they are irreversible reactions always, the reactions associated with the enzyme isomerases. We can see intermolecular shifting of the atoms, A with X here, B with Y, here it becomes A with Y and B with X. Like that, the intermolecular shifting of the atoms takes place. They are the different molecular species of the same enzyme. Those are the isomerases. And then last one, according to the previous International Union of Biochemistry classification, that is ligases. Ligases are those, they are also called as, this ligase is a Greek word, that is they synthesize, they are going to join together. Here you can see two substrates, A and B are separate, they are joining in the products. Those are called as synthetases or joining enzymes, ligases. And recently one more enzyme is incorporated here, that is the translocases. Translocases are those enzymes which are going to shift the molecules within the membrane, the transmembrane shifting of the atoms which are correlated or connected with the enzymes. Those are the translocases. Let us see few of the important oxidoreductases. We saw in our first session there are so many types of oxidoreductases. These redox reactions that takes place in the form of oxygenases, that is incorporate oxygen into the organic substrate, that is called oxygenases, catalyzing enzymes. And peroxidases, reduction of the hydrogen peroxide to hydrogen peroxide and also hydroperoxides, releasing the H plus and OH minus ions. And reductases, which catalyze reduction reactions. Dehydrogenases, oxidize substrates by transferring one or more hydride ions. And oxidases, oxygen as the hydrogen or electron acceptor. In these oxidases, oxygen is going to act as the H plus or electron acceptor. And then hydroxylases. Hydroxylases, they add hydroxyl group to the substrates. So either in oxidation or reduction of a particular substrate, different types of oxidoreductases can be seen. Then coming to the session 2, that is the chemical composition of the enzymes. We discussed in this particular chapter how exactly the enzymes are made out of. Enzymes are nothing but the globular tertiary proteins, three-dimensional structure. They are in their three-dimensional structure, they are very active. And we can see this is the three-dimensional picture of the enzyme. Enzyme primary structure will be like this, and when it gives a proper secondary structure, you can see the enzymes in the form of either alpha helices or in the pleated forms. And tertiary structure, we can see the joining together of alpha helix and the pleated structure. In between you can see heme and also beta polypeptides, that is the joining of the secondary structure, and also that gives the tertiary structure. This is the functional structure of the enzyme, wherein we can see most of the enzymes are very much active and functional. And quaternary structure can be seen, this is also seen in most of the enzymatic proteins. And also we discussed in the third segment, that is the property of the enzymes. Before going to property, I would like to tell a few things about the chemical composition. Enzymes are made out of proteins. So protein part of the enzyme is called as apoenzyme. Most of the enzymes which work better without the assistance of any non-protein part, non-protein substitutes. Those enzymes are called as simple enzymes. Enzymes which take the assistance of the non-protein the cofactor along with the apoenzyme or the protein part, those are called as conjugated enzymes. In conjugated enzymes, we tell apoenzyme plus cofactor gives us the holoenzyme or the functional enzyme. Without apoenzyme, only the cofactor cannot be functional, and also without the cofactor, enzyme is also waste. They are not going to act and produce the products. In the same manner, we also discussed cofactors, different types of cofactors, for example, organic cofactors and inorganic cofactors. Or amongst the organic cofactors, we discussed coenzymes and prosthetic group. Now, what are coenzymes? Coenzymes are nothing but those organic cofactors which bound to the apoenzyme very loosely. They can be easily separated from the apoenzyme part. Then we call such a cofactor as the coenzyme. In the case of inseparable, they are strongly bound to the apoenzyme, then we call such organic constituents as the prosthetic group. Most of the times, like NAD, FMN, FAD, along with that, the prosthetic groups can be inorganic ions, which are also tightly bound to the enzyme. For example, Fe++, Mg++, Mn++ ions, sometimes they are very strongly bound to the protein part or apoenzyme of the enzymatic protein. So they are called prosthetic groups. Sometimes the cofactor can be inorganic completely, and it is not tightly bound also. Such cofactors which are purely inorganic, they are simply called as the metallic ions, which are simply called as activators. Then coming to the third segment, that is the properties of enzymes. Enzymes properties can be distinguished into 6 major types. One is catalytic, another one is missing here, that is the colloidal nature of the enzyme, and then the sensitivity to pH, the heat sensitivity, and also specificity of the enzymes, and finally the reversibility of the reactions. In this order we studied in our regular sessions. And these are the properties. Quickly, if we glance, catalytic property, catalyst, like any other inorganic catalyst, the enzyme is also present in very small proportions. A very small proportion can act and give the products. And colloidal nature, colloids are nothing but the particles of dimension 1 to 100 nanometer, and they are suspended in the dispersion medium. In the same manner, the structure of the enzymes or the enzymatic proteins are dispersed, and they are going to have the collusion amongst each other, and that will facilitate in the enzyme action. The mechanism of enzyme action, we will study that in detail. And then coming to the sensitivity of the enzyme to very many factors like pH, heat, temperature. Like proper enzymes, for example, digestive enzymes, some are very much acid pH loving substances. That means they work better in the acidic pH, that is 2 to 4 pH, and some they are very active in basic pH. Likewise, the enzymes are highly specific to a particular pH. And then coming to the heat sensitivity, enzymes work better only under normal temperature, like normal body temperature 37 degrees centigrade. Until 45 degrees centigrade, enzymes work good, but that will be the optimum range of temperature above which the enzymes get denatured, they become non-functional. That is the heat sensitivity. And also specificity. based on the specificity, we have discussed in detail which are all the different types of specificity, like bond specificity or substrate specificity, and different types of specificities we studied. Please go through the session 3 for details. So enzymes are highly specific in their action. And finally, we discussed the reversibility of the reaction. Most of the enzymes show the reversible reaction. Sometimes the forward reaction is catalyzed by one enzyme and backward reaction the another enzyme. Sometimes most of the catalytic reactions are irreversible also. And all those with respect to reversibility of the reaction we discussed. And specificity, coming to the specificity, once again the enzymes are very specific in their action. A particular enzyme usually act upon a particular substrate, that we know that. And also they have exhibited these types of specificity: bond specificity, group, substrate, stereo specificity, geometric, and cofactor specificity. And also we discussed reversibility, sensitiveness to heat and inhibitors, and colloidal nature. Inhibitors also. See, they are highly specific to specific inhibitors. Inhibitors, competitive, non-competitive inhibitors. Competitive inhibitors are those which have the structural similarity with the substrate. So before the substrate come and binds to the active site, these competitive inhibitors can come and bind, and thus reaction will be stopped, no production of products. In the same manner, we discuss the vitamin properties of enzymes. How enzymes exhibit the properties which are similar to vitamins. Like enzymes are also organic substances, as do vitamins are. They are essential in very small or trace amounts. Vitamins also essential in trace amounts, and enzymes, requirement of the enzymes is also very, very smaller quantity for the maintenance of normal metabolism. They generally cannot be synthesized at adequate levels by the body. They must be obtained from the diet. Most of the vitamins are essential vitamins, like essential amino acids. Most of the enzymes are framed with the help of these vitamins. So vitamins and also derivatives of most vitamins, they act as the important coenzymes and cofactors. Thus, the absence or shortage of the vitamin may result in deficiency disease. In the same manner, the enzymes deficiency also causes disease, also impairs the metabolic activities. Then coming to the mechanism of enzyme action, we studied with respect to two famous models, that is one is Fisher's lock and key model, how exactly the enzyme will be having a specific grooves which are nothing but the active sites, which will conformationally or structurally they are going to be helpful for the substrate to fit into those grooves. When the substrate come and binds to the enzyme, exact bonding takes place. That is the transitional stage in the enzymatic reaction, that is the formation of the enzyme substrate complex. And later the enzyme will give out the products. There is a catalytic action, catalytic reaction of breaking the older bonds and making new bonds, and thus the substrate is converted into products, and enzyme is released without undergoing any chemical change. This is exactly resembling the lock and key. How exactly the key is, that is the substrate material, and the lock will be having a groove which is adjusting to that particular key. In the same manner, enzymes will be having the grooves which are called as active sites, to which the substrate come and binds. And next we studied another model to explain the mechanism of enzyme action, that is Koshland's induced fit model. Here what Koshland has told, enzyme will be there and substrates, and as soon as the substrate comes in the vicinity of the enzyme, they are going to create a certain conformational change. They induce the substrates themselves, they are going to induce the conformational change in the enzyme, so that they are making the active site suitable for them, and then they will bind and show the catalytic action and formation of the products. In both these models, one thing is common, that is the formation or framing up of the enzyme substrate complex. Okay, and also we studied how exactly the Arrhenius explains the phenomena of enzyme action. Arrhenius tells in all chemical reactions, as the reactants do have some kinetic energy, due to their kinetic energy, due to the collision among the molecules, a sort of energy is created. In the same manner, in any chemical reaction, without the absence without the enzyme, or in the absence of enzyme, the reactions can take place by increasing the temperature of the reaction mixture. So the mode of the formation of the reaction without the enzyme is this one, and the energy requirement for making the molecules to get the proper kinetic energy. Arrhenius tells some reactants will have the proper kinetic energy, so they can easily transform into products. But most of the reactants, they fail to possess the required amount of kinetic energy. They are called as low energy molecules. So these low energy molecules must be given some push. They must give some activation. So the energy requirement to make the molecules to acquire the required amount of energy, that energy is called as activation energy. Usually that activation energy, extra energy to channelize the low energy molecules, is called as activation energy. That activation energy is usually provided by increasing the temperature of the reaction mixture in vitro conditions. That means while working with the glass, or while doing any experiment, we can easily increase the temperature of the reaction mixture. That will facilitate the acquiring the proper amount of the energy, which is less in low energy molecules. That is the activation energy. The requirement of activation energy is very much high in the non-enzymatic reaction. We can see here, whenever the reactants are fed with the enzyme without increasing the temperature in the in vivo conditions, in the living organisms, what happens, the enzymes which are going to take up the boosting up action for the low energy molecules, this enzyme will do that function without increasing the temperature of the reaction mixture. So the requirement of this activation energy will be lowered in the presence of the enzyme. So activation energy requirement is very less. See, compared to the in vitro conditions, activation energy requirement in the in vivo conditions in the presence of enzymes is very, very low, and thus that will be easily converted into product. So change in this free energy, the enthalpy of the change in the free energy is quite interesting in case of enzymatic reactions. So that is how the progress of reaction with respect to free energy requirement is drawn in this graph. This explains as how exactly the enzymatic reactions takes place and how the enzyme is capable of accelerating the rate of the chemical reaction. And coming to the enzyme kinetics, the further study of how exactly the enzymatic reactions are going to give the special equation that tells us how the substrate concentration is very much important in any enzymatic reactions to acquire a specific amount of products. Okay, that we studied in the next chapter, that is enzyme kinetics. Enzyme kinetics is the study of the rate of enzyme catalyzed chemical reaction. The enzyme kinetics, the reaction rate is measured and the effects of varying conditions of the reactions are investigated. How in varied conditions the reaction rate will be differed. An enzyme, the E, is typically a protein molecule. We can see it is a protein molecule that promotes a reaction of another molecule, that is its substrate. So it promotes the reaction of the substrate. This binds to the active site of the enzyme to produce enzyme substrate complex and is transformed into an enzyme product complex, EP complex, and from there the product via the transition state, that is ES. The series of steps is known as the mechanisms. Okay, we can see here E plus S giving ES, E plus S, excuse me, I want to use this pen. Okay, E plus S gives ES and frames the enzyme substrate of the complex. That the intermediary changes once again to EP, that is enzyme product. Still the product is not detached from the enzyme, that complex, and then the products get detaches finally. So this graph tells us how exactly the substrate concentration is going to decide the mode of enzymatic reaction. This is rate of reaction. See, initially the substrate concentration, the intake of the substrate by the enzyme is quite high. It is going to increase, but at this particular point, that is the of Vmax, that point is the substrate concentration. If you keep on increasing, the active sites are completely saturated at this point, so there is no further scope for the substrates to come and bind. Then the Vmax, that is the conversion starts, begins, the conversion of the ES to the EP, and thus there is no rise in the substrate. The further rise of the substrate is not allowed. So the curve, initially it is increasing, now it is become steady state, and thus it is going to convert EP into the products. So what is this of Vmax? Of Vmax is 1, wherein the substrate the concentration increase is further no effect on the enzymatic reaction. That is, all the active sites are saturated at this point, and this of Vmax will tells us how much amount of the products can be possible. This is the deciding factor, which wherein the substrate concentration is done for this particular enzyme. No further substrate can be added, and only these many products are possible. So based on this basic idea, Michaeli-Menten they have explained how exactly of Vmax is going to decide the enzymatic reaction products. Okay, and also they explained the single substrate reaction and double substrate reaction. In the single substrate reaction, only one substrate will be there, so directly that substrate will be converted into product in one step after the formation of the enzyme substrate the complex. But later he also explained the double substrate reaction, that is the shifting of the enzyme from one substrate to another substrate. taking one functional group from the first enzyme and transferring that functional group to the second enzyme. This process is also called as ping-pong reaction. And how exactly the enzymes are going to help in the double substrates reaction. And Michaeli-Menten, they have given a universal equation. That equation helps us to determine the enzymatic reaction. Okay, that we discussed in our session. Quick revision, a quick glance on Michaeli-Menten's kinetics. What they are telling is how exactly the substrate concentration is nil, then we call it as V0. When we keep on adding the substrate till the active sites are saturated, the substrates will go on bind. That process till it reaches saturation, the curve will be increasing. When it reaches off Vmax, so it is going to decide the formation of the substrate. So in an enzymatic reaction, if the two substrates are there, A and B, then they are converting into ES, that is intermediary product. That reaction is taken care of with a velocity constant called as K1. And then conversion of the enzyme substrate complex to EP and finally P plus E. That reaction is catalyzed, or it is taken as the velocity constant K2. So the Km is the Michaeli-Menten's constant with respect to of Vmax. Km is equal to K2 plus K1 divided by K1. That is approximately equal to Kd. Kd is nothing but dissociation constant. That is dissociation of the enzyme substrate complex to the products. We can also put this Michaeli-Menten's constant in another way. That is Vmax is equal to Kcat E tot. The total amount of the enzyme required to catalyze the reaction with respect to the active, completely saturated active sites. This is about Michaeli-Menten's equation. We also studied Michaeli-Menten's phenomena with respect to Lineweaver concept and also Hopf's concept, wherein they have explained how exactly a linear curves can be interpreted to show that how off Vmax tells us the saturation of active sites and how it decides the enzymatic reactions. And in the next session, we discussed about the factors affecting enzyme activity. How temperature, optimum temperature is required for the enzyme action, pH, water, since most of the enzymatic reactions, most of the enzymes they are colloidal in nature, so they require the dispersion medium as the water. So most of the reactions, water itself is a substrate. So based on all these things, how water decides the enzymatic rate of the enzymatic reaction, and also concentration of the substrate. Just now we discussed in the Michaeli-Menten's equilibrium how the concentration of the substrate will be helpful in enzyme kinetics. And concentration of the enzyme, of course, the enzyme concentration, if you keep on adding the enzyme to the reaction mixture, definitely the rate of the reaction increases, because the inhibitors they become shorter to attack the enzymes, because we are increasing the enzyme concentration, so substrates can easily get the active sites, and thus the rate of the reaction is always enhancing in this case. And coming to the accumulation of end product, a very interesting factor, where we will see as soon as the end product gets accumulated in a biochemical reaction, there is no demand for that product till it is consumed again. So till that time, till it is completely exhausted in the living stream, Then during that time what happens, this end product itself will come and bind to the first enzyme of the sequential biochemical reactions of any metabolic step, and thus that is going to inhibit the activity of the first enzyme of the sequence, and thus the product making is stopped for the meantime till the product gets exhausted in the body. This enzyme, the first enzyme, will be shut down by the product itself. As soon as it is completely exhausted, then there is no product to shut the first enzyme, and thus the first enzyme again becomes active and the sequence once again continues. That is the accumulation of the end product, how it is going to regulate the enzyme action. And also in the next session we discussed the inhibition and also regulation of the enzyme action. Usually inhibition, we already discussed, it takes place with competitors. Competitors are of two types, that is competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are nothing but these are the competitors which are having the similar structural conformation with that of the substrates. Okay, competitive inhibitors, we call them as competitive inhibitors because they compete for the active site, since they resemble the same structure of the substrate. Before substrate go and binds to the active site, these competitors will go and bind, and thus no product is formed. Non-competitive inhibition is one wherein what happens, these competitors, they are not going to attack the active site, but they will attack the enzyme from some other place, and they will actually deform the enzyme, and the active site structure itself is deformed. You can see in this diagram, active site which is required for the substrate to bind is of U-shaped, but it is very much narrowed because of the reaction that this inhibitor has done to the enzyme. So it will attack the enzyme, it will enter the enzyme from some other place other than the active site, and it totally reduces the conformation, changes the conformation of the active site. Here it is narrowed down, it has reduced the groove that is required for the actual substrate to come and bind. That type of inhibition is called as non-competitive inhibition. And last one, that is allosteric inhibition or feedback inhibition. As I was telling, the lost product of the biochemical reaction, that itself is going to act as a controller or regulator of the first enzyme in the sequence. It will go and bind to the first enzyme. A very familiar example is for conversion of the L309 into isoleucine, which takes place in Escherichia coli, where we can see the first enzyme, that is the 309 dehydrase, is totally attacked by the converted product, that is isoleucine, the final product. Isoleucine will come and bind to the 309 hydratase till the isoleucine gets exhausted in the system. That is called as feedback inhibition or allosteric regulation. You can see the diagrammatic representation here. What happens is the product will come and attack the enzyme, first enzyme, that is the 309 dehydrase in the prescribed example there. So this allosteric, that is the feedback, that is the final product that is going to act as a allosteric activator once the isoleucine is exhausted in the mixture in the body. Okay, for example, what happens is this allo means different site other than the active site. This particular product or the feedback product is going to come and attach to the enzyme, and it will change the conformation of the active site. So the first substrate cannot bind to the enzyme, and thus that enzyme activity is completely stopped. Once the isoleucine is completely utilized by the Escherichia coli, then once again there is need for the production of isoleucine. At that time what happens, the same inhibitor or the regulator is going to change into allosteric activator, that itself acts as an activator, and once again it will correct the active site for the first substrate to come and bind. That is L309 once again binds to the 309 hydratase, and once again the sequential reaction continues till the formation of the isoleucine. Like that the feedback inhibition or allosteric regulation takes place. This completes our enzyme action and also enzyme chapter. And the next we started discussing about nitrogen metabolism in our next sessions. Let us quickly see what all we studied in nitrogen metabolism. Nitrogen metabolism, first topic is the sources of nitrogen. We all know atmosphere is made out of 78% to 80% of the nitrogen. The air is a mixture with maximum amount of nitrogen, and then comes the oxygen, nitrogen and other important gaseous mixtures. So nitrogen in the form of N2 or inorganic nitrogen is highly inert, and it cannot be assimilated, it cannot be taken up by the plant system. So it must be converted into some usable forms like nitrates, nitrites, ammonia, like that. And how exactly this conversion of the inorganic nitrogen to usable forms takes place, that we discussed in nitrogen fixation. Nitrogen fixation, mainly industrial or chemical nitrogen fixation, physical nitrogen fixation, biological nitrogen fixation. Under biological nitrogen fixation, involvement of the microorganisms will be there, and that too there are two types once again, that is symbiotic and asymbiotic. That means these microorganisms which are assisting in the nitrogen fixation can frame the symbiotic association with the host plant, or whether they are non-symbiotic. Like that we studied series of nitrogen fixation types. Before actually going to those, we can see the natural sources of nitrogen. For example, it is the natural sources, vegetarian, and these are the natural sources in nature, in the atmosphere nitrogen. And then atmospheric nitrogen, which is totally an inert gas. It is called as asphyxiant gas because it is toxic, and it also avoids the uptaking of the normal oxygen. So it causes the asphyxiation or suffocation, and even the organisms can die if there is complete blocking of the usable oxygen by this particular atmospheric nitrogen. So we can see natural source in rain plus industrial and vehicle emissions. Then oceanic nitrogen, the nitrogen from the Pacific Ocean. How exactly the nitrogen which enters the ocean and also water bodies as sediments, and also the leached out nitrogen products like nitrite and nitrate ions which enters the ocean body, and how they are again contributing the nitrogen source to the nature. And also waste water treatment plants, sewage plants, there also the sources of nitrogen can be seen. And also urban sources like fertilizers, vehicle emissions, like that, and marine sediments And they also have the near shore and deep water with nitrogen residues. And specific systems, they are also having the nitrogen as a source located in watersheds or during the shorelines. Along the shoreline you can see the nitrogen source. Then agricultural sources, we all know the fertilizers, livestock manure, nitrogen fixing crops, and also fertilizer application. All these are the sources of nitrogen that we use in agricultural sector. Likewise, the nitrogen sources are broadly classified into the inorganic and organic nitrogen sources. These are the organic nitrogen sources. Inorganic nitrogen sources are some of these. Okay, and also we discussed how atmospheric nitrogen will be always helpful in the conversion into usable nitrogen forms. So the conversion of the nitrogen from inorganic form to usable or assimilatory form is called as nitrogen fixation. I have two broad types, that is abiotic and biotic. Abiotic is one which takes place without the involvement of microorganisms, that is atmospheric or physical nitrogen fixation, which is natural because of the thunder and electric discharge, that is lightning. Due to lightning, how exactly the series of the changes takes place, utilizing the atmospheric moisture, nitrogen with oxygen gives the nitric oxide, NO. NO will again combine with O2, and it is going to give N2O and nitrous oxide, and also HNO2 and HNO3, and that reaches the soil in the form of acid rains. Likewise, the atmospheric or physical nitrogen fixation, and also we studied the industrial nitrogen fixation, that is Haber-Bosch process. It is purely a chemical combination of the nitrogen with hydrogen at higher pressures and higher temperatures up to 450 to 500 degrees centigrade, and pressure up to 300 atmospheres. And finally, very interesting nitrogen fixation part, that is the biotic with the help of symbiotic and asymbiotic organisms, microorganisms. Asymbiotic, if we name, there are aerobic bacteria like Azotobacter, anaerobic bacteria like Clostridium, and also blue-green alga, Nostoc, Anabina, etc. All these come under the asymbiotic nitrogen fixers. These they can fix the nitrogen without any symbiotic association. They themselves will be having in their filaments, for example Nostoc, Anabina, they'll be having the heterocyst cells. These heterocysts are having poor chloroplast content, and thus the photosynthetic rate is less, only photosystem 1, and there is no photolysis of water, so oxygen liberation is quite less, and that is going to help oxygen scavenging cell, what we can call this heterocyst cell, which are present in the blue-green alga, and thus the oxygen scavenging takes place naturally, which is quite responsible for the activity of the nitrogen reductase or nitrogenase enzyme. Hence, the nitrogen fixation can be easily taken up in these organisms without any proper symbiotic association. So these are called as asymbiotic nitrogen fixers. Then coming to the symbiotic nitrogen fixation, by making the root nodules and also shoot nodules. We can see root nodule forming nitrogen fixers are rhizobium and bradyrhizobium. Very importantly, bradyrhizobium is much more active when compared to rhizobium, because this bradyrhizobium can make association in a wide array of plants when compared to rhizobium. Rhizobium can make association only in leguminous plants. And also bradyrhizobium has a very huge multiplication capacity and also colonization capacity. That's why bradyrhizobium is much more efficient than rhizobium. Then coming to the stem nodule formation in azorhizobium, it is going to make the stem nodules. Let us quickly see the non-biological nitrogen fixation. Once again, lightning, these reactions takes place N2 plus O2 NO, that is nitric oxide. Nitric oxide again mixes with atmospheric oxygen to give nitrous oxide. Nitrous oxide clubs with atmospheric moisture to give hyponitrate and also nitrous oxide, nitrous acid. And NO plus water, it also gives nitrous acid and also nitric acid. So these two, when reaches soil, that is going to convert as acid rain, that is deleterious for the agricultural lands. And coming to the another type, the overall picture of the non-biological and biological nitrogen fixation, we can quickly see. Atmospheric nitrogen, that is the inert nitrogen with N triple bond N, this nitrogen in the atmosphere, when it is fed with the hydrogen, the industrial fixation, which is going to change into ammonia, utilizing lot of energy. So energy utilization is very high in the industrial fixation. And from there it is converted into nitrates by some nitrifying bacteria like Nitrosomonas, Azotobacter, like that. That is going to convert into nitrates and nitrites. And then nitrite is going to give nitrite to nitrate, and that is also possible through the lightning process. And once again it is denitrified, nitrates are denitrified by denitrifying bacteria back to atmospheric inert nitrogen, and some amount will be leached out to the sea and ocean, that will again be given back by the other sources. And we can see here biotic nitrogen fixation, wherein the fixed nitrogen enters the plants, and then these plant proteins and animal proteins, they are utilized by human beings, and all these organisms, when they die, again the decomposing bacteria and fungi, they are going to add up these nitrogen sources in the form of manure, in the form of fertilizer, that is with the decaying bacteria and also denitrifying bacteria. Once again the nitrogen stream will enter the atmosphere. Then in short, in brief, we can discuss this Haber-Bosch process, that is the industrial or chemical nitrogen fixation. Here nitrogen from the air is collected, and hydrogen from the natural gases or fossil fuels, that is fed together in very huge furnaces in the volume 1:3. 1:3 volume, they are going to add nitrogen and hydrogen at very high temperature, that is 400 to 450 up to 500 degrees centigrade, and atmospheric and pressure 200 to 300 atmospheres, in the presence of iron as catalyst. So they are going to have the untreated gases, they get recycled, and they are going to give the gases are cooled and ammonia turns to liquid, and then their framed ammonia will be stored and used for other industrial purposes. This is the very famous Haber-Bosch process in the 19th century, when there is need for the nitrogen. Lot of circus was going on at that time. Haber-Bosch process became the very important source of nitrogen formation. Then coming to the biological nitrogen fixation, we discussed non-symbiotic and also symbiotic nitrogen fixation. Quickly we will move on to the rhizobium as the important nitrogen fixing bacteria, which actually gave the formation of the nodules in the leguminous plants. How exactly the nodule formation takes place? Whenever the bacteria comes to the vicinity of the leguminous plants, they produces some lectins. At the same time, the root hairs also produces some flavonoids. These flavonoids will actually attract the bacteria to come and colonize near the root hairs. And once they get entered at this particular point, then the curling or the curling of the root hair can be seen. This is actually facilitated by the flavonoids, by the accumulation of the flavonoids. Then they fuse with the root cells and they get entry into the root cells. As soon as the root hairs they enter, they will make the infection thread. The infection thread is formed, and thus the bacteria will enter the cortical cells and they colonize there, and they are going to produce the special pigment, that is leghemoglobin, pink color pigment. This leghemoglobin, which is a very important oxygen scavenging substance, which is going to take up grab the oxygen. It will accept the oxygen and thus create the anaerobic condition which is congenial for the functioning of the nitrogenase. So nitrogenase enzyme is going to take up the further process. As the bacteria enters and they multiply in the cortical cells, they are called as bacteroids. These bacteroids will synthesize the nitrogenase, and as their volume increases, we can see the outgrowths in the form of nodules, that is the nodule formation. All these processes are triggered with the two different types of genes, that is NIF genes and NOD genes. These genes are going to promote these processes, the formation of the lectins and also formation of the flavonoids, and exactly the curling and formation of infection thread, and also the activation of the leghemoglobin hormone. All these are facilitated by the NIF and the NOD genes. Coming to the synthesis of amino acids. Amino acids are the important biomolecules containing amino and carboxylic group. There are so many essential and non-essential amino acids, but in plants all the amino acids are synthesized inside the plant itself. Only in the animals and human beings, of the amino acids amongst the 22 amino acids known in the genetic code, out of these 20 amino acids, nearly 22 proteinogenic amino acids were found in the plants. And we can see very importantly these essential amino acids are synthesized by two simple steps, that is reductive amination and transamination reactions. The amino acid synthesis pathway also we discussed, how exactly the three important metabolic cycles that of the respiration, that is glycolytic cycle, Krebs cycle, and pentose phosphate pathway. They gives the intermediary products of these cycles. They act as the precursor in the amino acid synthesis, and thus the amino acids are produced. For example, the aromatic amino acids like tryptophan, tyrosine, and also phenylalanine. These are the aromatic amino acids which are synthesized by taking the precursor materials, that is erythrose 5-phosphate and ribulose 5-phosphate of the pentose phosphate pathway. And also we discussed oxaloacetic acid and glutaric acid of the oxaloacetic acid and glutaric acid of the Krebs cycle, they are very much important in the production of the alpha ketogluteric acid. Those are the keto acids which are responsible for the formation of the glutaric acid. This glutamic acid is considered as the first acid in the transamination reactions. The series of transamination reaction by taking glutamic acid is the primary or initial amino acid. All other amino acids get synthesized, all the other 19 amino acids. And this transamination reaction is just transferring the amino group from one amino acid to another. And reductive amination is in the presence of the inorganic phosphate, the reduction of the one amino group from one substrate to another substrate. Likewise, we have studied the amino acid synthesis. They are very, very important since they are the building blocks of the proteins, hormones and enzymes. Then finally we studied the nitrogen cycle, which is a very, very important biogeochemical cycle. We already discussed during nitrogen fixation how this inorganic nitrogen is converted into organic forms or the usable forms, and they are taken up by the organisms, and again they will be given out to the environment in the presence of nitrification, denitrification and also mineralization processes, and we see the leaching, runoff and eutrophication, everything, all these processes, how the inorganic nitrogen is converted into organic forms, and once again they are given back to nature in the form of N2 or inert nitrogen gas. All these processes are important because there must be a complete balance with the flow of this organic and inorganic nitrogen forms. Otherwise, there will be imbalance in nature. Plants, they need the usable forms like nitrate, nitrite and ammonia forms. They are helpful in the production of the plant products, the agricultural sector. That's how we discussed the nitrogen cycle. That will be the last process, and the processes involved in the nitrogen cycle are nitrogen fixation, nitrogen assimilation, ammonification, nitrification and denitrification. And these are the common references you can go through for all these steps, that is for enzyme chapter and also nitrogen metabolism chapter. So this is the quick revision of these two major chapters of unit 1 of paper 8, plant physiology. All the best. Please go through the sessions in detail and prepare for your exams. All the best everyone. Take care. Happy learning.